Saliva has an enzyme called salivary amylase. Saliva has an enzyme called as salivary amylase. Salivary amylase enzyme has capacity to digest carbohydrates iodine test, carbohydrates in particular what type, starting from type of carbohydrates. So it's all the food stuff that we eat, like flour, ice flour, all this has starch. Okay? So when we discuss in first chapter, I think you already know iodine test. What is iodine test or starch test? When you put one drop of iodine on any starch substance like wheat flour, rice flour, etc. This iodine will turn into bluish black color or purple color. Bluish black color or purple color. Then when it turns into black or purple color, this will perform a starchy state. Okay?
So when iodine reacts with starch, it turns into bluish black or purple color. And we have discussed about this in photosynthesis chapter when we discussed about nutrition in plants. So carbohydrate is a type of starch which is present in wheat flour, potato, rice flour, all these. Okay. So the same wheat flour, rice flour or potato which has starch can be taken to perform this experiment with saliva. So once you need to confirm here, if starch is present, iodine turns bluish black color. If starch is absent, iodine will not turn into bluish black color. Okay, so for this, we are going to take 2 test tubes. For the experiment to prove the action of saliva, we are going to take 2 test tubes. This is test tube 1. Test tube 1. This is test tube 2. Test tube 2. In first test tube, I am taking a solution, water plus rice or water plus wheat, something like that. Here also we'll take water plus rice or water plus wheat, something. Same whatever we have taken in first test tube, second test tube also will take the same. Okay. We'll leave it for a few minutes, 1 or 2 minutes. After that, we'll add to the first test tube, we'll add few drops of saliva. So here saliva is added. Here saliva is not added. Saliva is not added. Okay. We'll leave it for something around 20 minutes. After that, we'll perform iodine test. Okay. So when this test tube is added with iodine drops, will it turn into bluish black? Or if this test tube is added with iodine drops, will it turn into bluish black? Which one do you think? Since wheat is present already, it should turn into bluish black. But what is happening? The saliva which is added to wheat will break the wheat. Whatever starch that is present in wheat will be broken down into simple molecules. Simple molecules. So these simple molecules that are formed from starch cannot turn iodine drops into bluish black color. Okay. So that is the reason why in this there is no bluish black formation, no bluish black color formation, but here in this solution we can see bluish black color formed. Bluish black color formed. Okay, we'll summarize this once. I'll say you. You can do this experiment if you have iodine at home. So what we are doing, wheat plus water plus saliva. What will happen? Carbohydrates, that is nothing but starch, is broken down into simple molecules. So iodine test fails. Why iodine fails? No starch. If iodine test does not show bluish black color, it is failed. It is not turning into bluish black. Why? No starch. Starch is converted into simple molecules. When you take wheat plus water plus only iodine drops. Okay. Here also we added iodine drops. Here also we added iodine drops. But this iodine test succeeds. Why? Starch is present. Starch is present. Okay. Where starch is present? In wheat. Here what happened to the starch? It broken down into simple molecules. Okay. That is the result of the test. Next, after this, next one, nutrition in amoeba. Nutrition in amoeba. In nutrition, amoeba nutrition also, we can see those following steps which we discussed. Okay, you have some doubts. I'll answer you at the end, ma. Nutrition in amoeba. Nutrition in amoeba has those following steps which we discussed in human nutrition. Ingestion, digestion, absorption, assimilation, and finally defecation. So in amoeba also these all steps can be seen. When an amoeba finds any food particle near, it extends its pseudopodia, false feet-like structure towards that food particle, and this pseudopodia come close like this. They come close like this, and gradually these two plasma membranes will fuse together to form a vacuole here. This is a vacuole that is formed. It is nothing but food vacuole. This food that is present inside the amoeba gradually gets digested. Okay. Once it's get digested, the food particles will be absorbed into its cell contents. After it is entering into cell contents, it is used in the synthesis of structures like cell organelle, cell membrane, different structures inside the cell. That is called as assimilation. After that, this food vacuole will not be present. After the food is digested, the food vacuole will be Also, it will be disintegrated. Gradually, it will disappear. Now here waste materials are present. All the waste materials that are present in amoeba's body. If food is getting digested and accumulation, definitely some amount of waste will produce. All that waste will accumulate in one area from this all parts. And once it is accumulated in one area, amoeba opens itself and leaves out some amount of that waste material by the process called defecation. So this is the nutrition amoeba. We'll see the diagrammatic representation of this. Amoeba and food particle. Amoeba extended its foot-like structure called pseudopodia around the food particle. And slowly what will happen? Amoeba forms is the cytoplasm comes close and the food particle will be enclosed inside. Next, what happens? This is food vacuole. Food vacuole. After the ingestion is completed, this is nucleus. This is food vacuole. The food particle will slowly be digested. After digestion, amoeba slowly absorbs all the nutrients in its body and assimilates. Okay, next. So this parts will be absorbed into the body and they get assimilated. They are used for formation of different structures. As a result, waste material will be produced. This waste material that is produced will accumulate at one point and finally this waste will be thrown out of the cell body. Okay, so this is first, second, third, fourth, fifth, sixth, seventh, eighth. 1, 2, 3, they are related to ingestion. 4, digestion. 5, absorption. 6, assimilation. 7, 8, defecation. Okay. These three are ingestion, this is digestion, this is absorption, this is assimilation, and these two are egestion. Egestion or defecation, same thing. Throwing out the waste material or undigested food material from the body. So this is the nutrition in amoeba. Now let us talk. So I'll write that 1, 2, 3 of the previous diagram. Ingestion, 2, digestion, 4, digestion, 5, absorption, 6, assimilation, and 7 and 8 are defecation. Okay. So let us see this steps that are involved in paramission. Digestion, food nutrition in paramission. This is paramission. Total body of paramission is covered with cilia-like, hair-like structures called cilia. So these cilia that are present over the body of the paramecia not only help in movement. One of the major function of the cilia is it helps the paramecia to move. Just like how the people in boat move with the help of oar, this cilia will push the water one side and this paramecium will try to move forward. So you can guess the movement of paramecium, how much slow it will be. It's very slow movement. Okay. So this is the structure of paramecium, basic structure. So this part of the paramecium is called as cytostome. Cytostome means mouth. Whenever it finds any food particle, this paramecium pushes the hair of the paramecium, cilia of paramecium, bring this food particle close to its cytostome. After that, this food particle enters inside. When it is entering itself, the food vacuole is formed around it. Okay, this will convert into this structure. Once the food vacuole is formed, the next step is the food that is present inside this will be slowly digested. The digested food slowly gets absorbed into the paramecium body. After it is absorbed, paramecium accumulates all those nutrients that are obtained in various parts of the cell. Next, after that, this food vacuole takes in all the waste material. Waste material will be collected, and at any point in the plasma membrane, it makes a small break and throws all this material outside, waste material. This is called as egestion, and the pore that is formed is called as anal pore. The hole that is formed in the cytoplasm plasma membrane is called as anal pore. So what are the steps involved here? See, this is nothing but, I'll write numbers here: 1, 2, 3, 4, 5. First one is ingestion of the food. Second one is digestion. Third one, absorption. Absorption is followed by assimilation, but it cannot be shown separately. In our body also, can we show assimilation? So it's not possible for every organism to show assimilation. So in fourth step, what is happening is collection of waste material and defecation. 4 and 5. Defecation. Undigested food material from various parts is collected into that food vacuole and it is thrown outside. Okay. Is it clear, children? So that is the mode of nutrition that is seen in paramission.